So good morning, grade 12 students. In this session, we're going to continue what we have started before about the immunology introduction. And in this session, we're going to continue what we have started in last week, and we'll continue from phagocytosis. We talk about phagocytosis. We said that the phagocytosis is the non-specific immediate immune response. What the meaning of non-specific? Meaning that it doesn't depend on the identity of the intruder, on the identity of the non-self. In addition to this, immediate immune response, meaning that it starts immediately, instantaneously, and it doesn't wait for some time to start. The main actors are for the phagocytosis, they are the phagocytes. What do we call them? They are the monocytes and the granulocytes as well. And the phagocytosis step, first the phagocyte will approach toward the pathogen. It approaches toward the pathogen. In this figure, we have the macrophages coming near the bacteria. Then the second step, the phagocyte will adhere to the pathogen and gets in direct contact with it. And then the third step, the phagocyte will engulf, ingest, and digest the pathogen. These are the three important steps for the process of phagocytosis. So as I said, the main actors of the phagocytosis, they are the phagocytes, they are the monocytes and the granulocyte. Monocyte, they have pores who shape the nucleus, and granulocytes, they have multiloopy nucleus. We talk about the clotting of the blood, how does the clotting occurs, and the steps. Now, moving for the specific immune response. Let's remember together what are the lines of defenses. The first line of defense, if you remember, they are the natural barriers, the skin, the nasal secretion, the gastric juice, the sweating, the urine in the urinary bladder or in the urinary system. All of them, they represent the first line of defense. What happened? The first line of defense is broken. We can move to the second line of defense. Second line of defense is the non-specific immune response. Non-specific immune response, meaning that we have the inflammation and the phagocytosis. What happened if this line was also broken? We move now to the third line of defense. Third line of defense, it represents the specific immune response. First two line of defenses, they fail, so we move to the third line. As I said, first line of defense is the natural barriers. Second line of defense is the non-specific immune response. Third line of defense is the specific immune response. Let's start with the cases of activation of T lymphocytes. Remember when we talk about the T lymphocytes, they have large rounded nucleus, they have a small cytoplasm, they have a special receptor, they are called TCR, T cell receptors. And also the T lymphocytes, if you remember when we talk about them, we said that the T lymphocytes, they are called the killer cells. They kill other cells using the TCR, using their T cell receptors. Cases of activation of T lymphocytes. One, the T lymphocytes, they are activated. T lymphocytes, they are activated first whenever we have a viral infection by a virus. So the T lymphocytes, they are activated. Second case of the activation of T lymphocytes is the case of graft rejection, and in specific, in the case of allograft. So we have two cases for the activation of T lymphocytes. First case is the case of the viral infection by virus, and the second one is the graft rejection in the case of allograft mainly. So what are the steps of the activation of T lymphocytes? Steps of activation of T lymphocytes, the specific T lymphocytes. Here, I'm talking about the specific immune response. So here it depends on the identity of the intruder. In the T lymphocytes, they are activated in two cases. First, whenever we have a virus. Second, whenever we have an allograft or graft rejection. So they are specific for these two cases. First, the specific T lymphocytes, they approaches toward the target cell or the infected cell. Second, the T lymphocytes, they adhere or they bind to the target cell, they attract the target cell and they bind to it using their T cell receptors. Using what? using their TCRs, T cell receptor, using their TCRs. So they get indirect contact. First they approach, then they get indirect contact, and the third step, what will happen? The third step, the T lymphocytes, they give the target cell. So they get indirect contact with it, then they give it a killer kiss. This killer kiss will cause perforation. What the meaning of perforation? Will create holes. Yeah, it will make holes inside the cell. These holes will lead to the death of the cell and its end. So here, the T lymphocytes, they give the target cell a killer kiss. This killer kiss will cause perforation, meaning that it causes holes inside the cell. Imagine that we have a sac, this sac containing water. I cause, using a pen for example, a minute holes inside this. So the water will go out of the cell and the component of the cell will go out of it, and this will lead to the death of the target cell. This is called the killer kiss. So T lymphocyte has three important steps for activation. First, the specific T lymphocyte approaches toward the target cell. It's very similar to the steps of phagocytosis, unless in the last step. And in the second, they use TCR. So first, approach, get indirect contact and bind to it using TCR, and then they will give the target cell a killer kiss. So here, approaches toward the pathogen, gets indirect contact with it, and then here giving her a killer kiss, and this is the cancer cell, by the way. Here, type of the immune response carried out by T lymphocytes. All of you know that the T lymphocytes from now on, they carry a specific immune response. What about the name of the response carried out by T lymphocytes? The name of the response carried out by T lymphocyte is called cell-mediated immune response. So cell-mediated immune response. So from now on, T lymphocytes, what do? They carry out specific cell-mediated immune response. Cell-mediated specific immune response. So here, specific, you know that T and B, they carry out specific immune response. Cell-mediated is only for the T lymphocyte. So specific cell-mediated immune response. When the T lymphocytes, they are the active cell, or they are the major cells that work in this response. What do? They kill the cells using what? The TCR. That's why they call it TCR, T cell receptors, and that's why they call the T cells as killer cells, because they kill the other cells using TCR. Now, let's talk about the blood. All of you know that we have different blood groups. Different blood groups, we have how many different blood groups? We have 4. We have blood group A, blood group B, blood group AB, and blood group O. Remember that whenever we talk about the blood cells, we said that the red blood cells, they don't have a nucleus, as well as they don't have HLA on their surface. So here we have blood group A. Blood group A, it has antigen A. Blood group B, it has antigen B. Blood group AB, it has both antigens A and B, while blood group O, it has no antigens. From now on, antigen is the same as the blood group. So blood group A has antigen A, blood group B has antigen B, blood group AB, it has both antigens A and B. However, blood group O ما عندنا شيء اسمه antigen O, so it has no antigens. However, blood group A, blood group B, blood group AB, and blood group O. I think you heard about agglutination. So what is agglutination and how does agglutination occur? We're going to discover this in the coming slides. So first, you have to know that the blood group contains the antigens that are found on the red blood cells, the same as the blood group. So blood group A, it has antigen A. blood group B antigen B. blood group AB ما في عندي antigen اسمه antigen AB. عندي antigen A and antigen B. Both of them. We have two antigens. And blood group O has no antigens. So what about the antibodies? Antibodies, they are different from the blood groups. So let's start step by step. And the first blood group A. Blood group A, it has on the surface, it has antigen A. Whereas in the serum, we have antibodies. So from now on, antigens, we gave them the symbol Ag. Antibodies, ABS. So antigen is the same as blood group. So blood group A, antigen A. Antigen A should not be present with the same antibody. So we can't have antibody A. Why? Because they will attack each other. So here, antigen A, the antibody in the serum will be opposite to antigen A, which is antibody B. Always the antigen is the same as the blood group, while the antibody is the opposite of the blood group. So antigen A, antibody B. However, blood group B has antigen B, while the antibody is different from antigen B, it's antibody A. Blood group AB has both antigens A and B. However, we can't have antibody A or antibody B because if you have them, they will attack each other. For example, antibody A will attack antigen A, and antibody B will attack antigen B. So here, we can't have the same antigen and its corresponding antibody in the same blood group. So here, blood group AB, it has no antibodies. However, if we move to blood group O, it contains no antigens. But blood group O contains antibody A and antibody B. So here, both of them, they are present. As you see here, the blood group has an antigen and antibody. So antigen A is the same as the blood group, always the antigen, the same as the blood group, while antibody is totally different. It's the opposite of the blood group. Let's continue with antigen. As I said, is the symbol Ag. It has another name. Sometimes we're going to see it as agglutinogen. So agglutinogen is the same as antigen, while antibody is agglutinin of symbol Ab. To study the possibility of transferring blood from a donor to a recipient. We talk about donor and recipient. We said that the donor is the one that donates. Recipient is the one that receives. So we have, in order to study the agglutination, we have to know what is the antigen or the antigens found on the red blood cells of the donor. So always, the donor, we have to look to the antigens. So antigens of the donor. Always and always and always. Donor, look to the antigen. Recipient, you have to look to the antibodies found in the serum. So always donor, I have to look to the antigen. Recipient, I have to look to the antibody. Third step, no agglutination should occur. What is agglutination? Agglutination is the binding of the antigen with its corresponding antibody. What does this mean? This means that if we have blood group, if we have, let's say, antigen A and antibody B between the donor and the recipient, antigen B and antibody B. Antigen B and antibody B, whenever they found in the same blood group, they will fight and they will cause a reaction called agglutination. In this case, the blood cells, they are going to be broken and this blood transfer is not accepted. So here, let's have an example. Who's here in the live session? Who's blood group A? في حدا منكم blood group A? Blood group A? Anyone is blood group A?
Me?
مين blood group A؟ في حدا blood group A؟ Hassan, انت blood group A?
Yes.
Okay. So Hassan, let's have an example about Hassan. في حدا blood group B? blood group B. في حدا منكم blood group B? Noura blood group B? Okay.
Yes.
So Hassan and Noura. So Noura needs blood, but the blood. فحسن بده يتبرع بالبلاد حسن. Okay? So قلنا Hassan blood group A. Noura is a blood group B. Let's have this example. So Hassan always he's the donor. So أنا ما بدي أحسب هو donor or recipient. Always blood group A. أي antigen بيكون عندك يا حسن؟ قلنا الانتيجين is the same as the blood group. So أي antigen عندك يا حسن؟ Yes?
A.
Antigen A. Very good. بينما الانتيبودي is opposite to the blood group. So شو بيكون antibody? B. Okay? نورا blood group B. نورا الانتيجين اللي لها بده يكون the same as the blood group. So عندها antigen B. بينما الانتيبودي بده يكون opposite. يعني عندها antibody A. هلا everything is good? Yes محمد. Question?
طيب blood group O.
انت blood group O وهلا بنحكي about blood group O. يلا خلينا نخلص this example and then we can move to blood group O. No problem. So here عندي blood group A and blood group B. تمام؟ We said that donor. شو انا بدي اطلع يا حسن on the donor? Antigen or antibody? Donor. We have to look to the antigen. So antigen حسن عنده antigen A هو الدونر. So أنا بدي أطلع له على الانتيجين. Wherever نورا she's the recipient. هي الريسيبينت. أنا على شو بدي أطلع للريسيبينت؟ To the antibodies. So شو عندها نورا antibodies؟ عندها antibody A. antigen A وantibody A. يعني في عندي for example الانتيجين والانمي تبعوله وعدوه. انتيجين A والانمي تبعوله اللي هو antibody A. So شو هيعملوا؟ هيعملوا fighting with each other. So هيعملوا شيء اسمه agglutination. So هيتخانقوا مع بعض ويعملوا agglutination. This reaction, whenever it occurs, meaning that this blood transfusion is not possible at all. مستحيل إن تصير هيدي blood transfusion. مستحيل blood group A يعطي blood group B. This is impossible. مستحيل إنها تصير. مستحيل blood group A يعطي blood group B. Why؟ لأنه blood group A هو الدونر عنده antigen A وبلاد جروب B هو الريسيبينت عنده antibody A. So antigen A وantibody A يتخانقوا مع بعض. شو هيعملوا agglutination. So here the blood cells will be for example destroyed. أكيد ما حتصير. هيدي إذا صارت إذا بحال صارت بيجي مستشفى this blood cells they are going to be destroyed. إذا ما لحقوا عنده high risk for death. نيجي لبلاد جروب O اللي عم بيحكي عنه محمد. أنت محمد بلاد جروب O؟ بلاد جروب O. طيب هنشتغل بلاد جروب O اللي هو محمد. So زياد بلاد جروب O. وخلينا نشتغل بلاد جروب اي بي اللي هو في حدا بلاد جروب اي بي هون؟ ما حدا بلاد جروب اي بي؟ طيب انا مستر محمد بلاد جروب اي بي. سو مستر محمد انا بلاد جروب اي بي. اوكي؟ سو مستر محمد هو بلاد جروب اي بي. مين بدك يكون الدونر يا محمد؟ مين بدك؟ انت تكون الدونر ولا انا اكون الدونر؟
انا الدونر.
انت الدونر. اوكي. سو بلاد جروب او هو الدونر والريسيبينت هو بلاد جروب اي بي. هلا عم نحكي about blood group O blood group AB. انا اصلا حطيت O مع AB لانه بدي احكي about O and AB they are special. So blood group O يا محمد شو عنده antigens؟
او
عنده antigen O. ما في antigen O نهائيا. في عندي antigen A وantigen B بس.
انتيجين بي.
ما عنده لا antigen A ولا antigen B. So blood group O ما عنده no antigens. اوكي؟ بينما antibodies عنده الاثنين. عنده antibody A
antibody B.
antibody B. تمام لهون؟
مم.
اوكي. Moving to Mr. محمد عنده blood group AB. شو يعني عنده أي antigens؟ عنده
أي
antigen
بي.
A وانتيجين B. مش antigen AB. Pay attention to this. لأنه في كثير منهم بيقولوا antigen AB. ما في antigen اسمه antigen AB. في antigen A and antigen B. So here عنده اثنين antigen A and antigen B. هون antibodies ما عنده. لأنه إذا عنده واحدة من الانتيبوديز حيعملوا agglutination. So ما عنده no antibodies. قلنا الدونر يا محمد عشان نطلع له دايما
على الانتيجنز.
عن انتيجين. شو عندك انتيجينز؟
ما عندي.
ما عندك. وانا شو عندي انتي بوديز؟ ما عندي.
ما عندي.
So this is possible or not؟ ممكن تصير هيدا blood transfusion or not؟
نو.
مش ممكن تصير؟
إذا ما في انتيجن ما في انتي بودي.
يعني مش ما حيصير
ما
في
حيصير
agglutination.
اجلوتينيشن.
يعني no agglutination. يعني this one
يعني
is
في
possible.
اتس possible.
يعني this one is possible. This one is possible 100%. هلا بقول لك ليش. No agglutination. So here is possible. يس حسن سؤالك قبل ما كفي my idea. يس.
اذا الاي بي الدونر بس
إذا
فيهاش
اي بي
تعطي
دونر هلا حاحكي عنه. يلا بس اعطيني few seconds. اوكي حسن؟
اوكي
اوكي. طيب محمد زياد بلاد جروب او. بلاد جروب او ما عنده انتيجنز. يعني مع اي بلاد جروب ما حيعمل اجلوتينيشن لانه ما في عنده انتيجنز لحتى يعملوا اجلوتينيشن. سو بلاد جروب او بسموه له اسم بسموه يونيفرسال دونر. ليه يونيفرسال دونر؟ انا على الدونر على شو بتطلع اتفقنا؟ بتطلع على الانتيجنز. سو هو ما عنده انتيجنز. سو نحن بنسميه يونيفرسال دونر لانه ما عنده انتيجين في يعطي الكل. سو بلاد جروب او هو اكرم واحد في يعطي الكل. في يعطي بلاد جروب A، بيعطي بلاد جروب B، بيعطي بلاد جروب AB، وبيعطي بلاد جروب O اللي مثله. So he's called the universal donor since it can donate the blood group for all the blood groups. However, blood group AB بسموه universal recipient. ليه بسموه universal recipient؟ لأنه في ياخذ blood group من الكل. ليش؟ نحن قلنا الريسيبينت عشان بنطلع له؟ بنطلع له على الانتي بوديز. فبلاد جروب اي بي ما عنده انتي بوديز. يعني هو في ياخذ من كل بلاد جروبس بس ما بيعطي حدا. سو بلاد جروب اي بي اللي هو مستر محمد هو بأبخل واحد. ياخذ من الكل بس ما بيعطي حدا الا اللي مثله. However blood group O هو اكرم واحد. هو بيعطي الكل بس ما بياخذ الا من اللي مثله. So that's why it's called the universal donor and the universal recipient. So blood group O universal donor لانه ما عنده انتيجين. ما حيعمل agglutination مع اي blood group. وuniversal recipient blood group AB لانه ما عنده انتي بودي. يعني كمان ما حيعمل agglutination اذا اخذ من اي blood group. يس حسن سؤالك كان حبيبي شو هو؟
مش الحال.
مش الحال؟ اوكي yeah. So let's continue the idea about the blood groups. محمد عندك سؤال قبل ما نكفي؟
استاذ يعني يعني الاو ما بياخد الا من الاو.
او ما بياخذ الا من او. هلا ما بدي فوت بوزيتيف ونيجاتيف بنحكي عنهم بعدين. اوكي؟ اذا او بوزيتيف او نيجاتيف اي بي بوزيتيف اي بي نيجاتيف نحكي عنهم بعدين in the coming sessions. تمام؟
اوكي.
Okay, yeah. So may a person of blood group O donate a blood to blood group person of blood group B. already احنا جاوبناهم ثلاث ارباعهم. يعني blood group A شو حيكون عنده انتيجين A، وبلاد جروب B حيكون عنده antibody A. فمستحيل يصير حيصير في عندي agglutination. So هذول questions already we discovered them in this session. So 4 persons in the lab whose blood groups we want to determine. يعني عندي 4 اشخاص باللاب بدي اعرف هيدول شو البلاد جروب اللي لهم. So اخذوا سامبلز. انت بس تروح على اللاب كيف بيعملوا بلاد جروب بلاد سامبل بياخذوا نقطه بحطوها on the slide 3 نقط وبيعملوا لك سامبلينج عليها. They add antibodies to each drop. يعني بيزيدوا antibody A antibody B بيزيدوا شيء اسمه ريسس antibody ريسس. يعني بيزيدوا اذا هي بوزيتيف او نيجاتيف. هلا بنحكي عنها. So samples of these blood were obtained on different sera. Sera هي the plural of serum. Serum قلنا وهي containing antibodies. So بس نحكي عن serum we are talking about antibodies. So بس نحكي عن serum عم نحكي عن antibodies يعني أي cell بتعمل secretion for the antibodies of the B lymphocytes. عم نحكي عنها in the coming slides. So the results are summarized in the following table. هيدي الريزلتس خدنا معنا 4 samples زدنا لهم antibody A و antibody B. هيدي اللي فيها dots مثل مغربلة هيك هي for agglutination واللي هي red ما في عندي agglutination. So sample 1 لما زدت له يلا step by step رح تحلوا معي هلا يلا. So sample 1 زدت له antibody A. إذا زدت له antibody A عمل agglutination يعني أي أنتيجين عنده. يس أي أنتيجين زدت له antibody A عمل agglutination. antibody A مع مين بتعلق مع أي أنتيجين؟ أكيد حتعلق مع الأنتيجين اللي مثلك. So حتعلق مع أنتيجين A. يعني هيدا عنده أنتيجين A. زدت له antibody B عمل agglutination يعني عندي أنتيجين B. So عنده أنتيجين A وأنتيجين B. شو بيكون البلاد جروب للسامبل 1؟ بيكون البلاد جروب هو A B. مشي حالنا أول وحدة؟ راما مشي حالنا؟ راما hello it's me.
مش الحال.
Okay علي حريري second sample sample 2. We added antibody A to sample 2. عندي agglutination.
In sample 2, when we added antibody A, there's no agglutination.
يعني عندي antigen A or not?
لا ما في antigen.
No antigen A. While when we add antibody B,
there is agglutination.
يعني what do we
So
have?
there is agglutination.
يعني عنده بس antigen B هيدا البلاد جروب blood sample. شو بيكون البلاد جروب له؟
Blood group B.
blood group B. Excellent. Sample 3 زدنا لها antibody A عملت agglutination يعني عندي antigen A. بس إذا زدت لها antibody B ما عملت agglutination يعني no antigen B. يعني شو البلاد جروب حيطلع؟ حيطلع البلاد جروب هو A. آخر وحدة no agglutination with antibody A ولا antibody B. يعني ما عندي لا antigen A ولا antigen B. So what is the blood group? The blood group is O. هيك بيعرفوا باللاب what is the blood group and how can we determine this. So interpret the obtained results to indicate the blood group of each person. May person 1 indicate to person 2. هيدول اللي حكيناهم. After adding antibody A to antibody B to sample 1, agglutination صارت both cases. What does this indicate? This indicates the presence of antigen A and antigen B in the sample. So person 1 is blood group AB. ثاني واحد زدنا antibody B A antibody B صار عندي agglutination بس in the case of antibody B يعني عنده بس antigen B so it's a blood group B. لما زدنا antibody B and antibody A to blood sample 3 agglutination صارت مع antibody A بس. So here we have only antigen A. However لما زدنا antibody A and antibody B to sample body 3 blood 4 sorry. So here we have no agglutination in both cases. So ما عندي لا antigen A ولا antigen B. So the blood group is O. So the donor blood group AB. So عنده antigen A and antigen B. دايما بنطلع على الدونر على الانتيجين. recipient هو blood group B. So blood group B شو بنطلع له recipient على الانتي So عنده antibody A بالسيروم تبعوله. So هيدا السيروم عنده antibody A حيعلق مع مين؟ حيعلق مع الانتيجين A. هيدا الانتيجين A والانتيبادي A حيعلقوا مع بعض. حيصير في عندي agglutination. So this blood transfusion is not accepted. So cases of activation of B lymphocytes. امتى بينعمل لهم activation البي؟ احكي لك T in the viral infection or in the case of graft rejection. Activation of B lymphocytes case of the blood transfusion. ليش blood transfusion؟ لأنه بصير عندي secretion of antibodies. And the second case of bacterial infection. لما يكون عندي infection by a bacteria. So here a green antibody it's fighting against an antigen. Be careful. Every antibody is specific for an antigen. So antibodies, they are specific. هنحكي عنها in details in the coming sessions. So what is the type of the immune response carried out by B lymphocytes? Is the specific immune response. Type of the immune response who is specific, name هو humoral immune response. So B lymphocyte شو اسمه immune response؟ اسمه specific humoral immune response. مين الactive فيه؟ in the B lymphocytes. What do? They secrete antibodies. Where? Inside the serum to kill the non-self. So أنا بس أحكي about serum أنا عم بحكي about antibodies. شو هو السيرم؟ أنت لما تروح على المستشفى بعلقولك شو؟ بعلقولي مصل. هيدا المصل هو السيرم. بقلبه antibodies. So أنا بس أحكي antibodies عم بحكي B lymphocytes يعني الاميون ريسبونس is specific humoral immune response. بس احكي T lymphocytes عم بحكي specific cell mediated immune response. So characteristics of the specific immune response. شو characteristics? Characteristics عندي شيء اسمه primary عندي شيء اسمه secondary. Primary immune response and secondary immune response. Primary immune response. What is the immune response primary and what is the secondary? Primary immune response is the response upon the first contact with the pathogen. يعني أنا أول مرة بشوف الفلو فيروس هي بنسميها primary immune response. However, second time I saw this is called the secondary immune response. If the immune response upon the second contact with the same pathogen. يعني أنا شفت الفلو فيروس أول مرة جربت شي 6-7 أيام. شفته تاني مرة بسميها secondary immune response. جرب 3-4 أيام. Why? This is the important question that we are going to answer it in the next session. So primary immune response, secondary immune response, difference between these two, why the primary immune response is, for example, takes a longer time, and why the secondary immune response is faster and eliminates the pathogen in a faster way. We're going to see this in the next session.